Yeah, learning objectives, just want you guys to understand what our software requirements are for customers in order to operate our instruments, and it's going to be different for Onso and our long read platforms. Um, know when to initiate customer conversations about software throughout the sales cycle so that we don't catch anybody off guard about certain requirements that they'll need to do their sequencing. We're going to talk a lot about a piece of software called SmartLink, which is very central to our long read platforms in order for you to um, connect to your instrument, uh, operate the instrument setup runs, monitor runs, and look at sequencing data. So yeah, a lot of slides on that. And then just a brief guide on how to inform customers about our secondary and tertiary data analysis, and I'll provide some definitions on primary, secondary, and tertiary later in this deck. Okay, so I've put together this slide. This has evolved over time, but just a very quick, very high-level overview of our different sequencing workflows. Long and short reads have the technology for actually doing the sequencing is very different, but if you look at it from like a high-level workflow perspective, they're actually fairly similar. They share the same high-level steps. It's just the details, specific details about them are very different. So starting up with like library prep, there's a library prep step for both long and short reads. For long reads, you will create these smart bells. On short reads, you'll ligate your adapters at the ends of whatever genomic insert you're trying to sequence. That's all on the wet lab side and at the bench. Then when you're ready to go start actually sequencing what you've prepped, you have to you have to set up a run, and that's basically just telling the sequencer, here's all the settings I want you to run my sequencer with. On long reads, you're going to be defining things like movie times, like I want to run 24-hour movies or 30-hour movies while it's sequencing. Because it's sequencing in real time. On short reads, it's taking snapshots of the flow cell at certain points in time. And so you'll be defining things like number of cycles, like is this going to be a 150, 2 by 150 run? I guess it'd be, I guess the technically correct terminology is like 150 by 150 if you're doing paired end. How many indexes am I going to, how many index bases am I going to sequence, things like that. Also defining the barcode sequences that you're attaching to each sample that you've loaded into your sequencer so you can demultiplex them later into separate files. Then you actually do the sequencing itself while it's in, you just load it onto the instrument, the Revio up here, and this is what the Onso looks like down here. And then from that point on, it's pretty hands-off, right? Like the sequencer kind of does all the work, but then so on the long read side, there's a base caller that's running on instrument, looking at the polymerase binding nucleotides and fluorescing in real time. And so there's a whole algorithm on board that's taking care of that. On short reads, you're looking at snapshots or images of clusters being fluoresced on the flow cell, and then algorithm looks at those fluorescences over time and calls a base based off of the color of the dye. And then after you get all the base calls, then you run demultiplexing and regeneration, putting together all the bases that were called into reads and then splitting them out into separate samples. And that's splitting out into separate samples is what we call demultiplexing. And so the step itself is pretty similar between the two, but the outputs you get are different. So on long reads, we output what's called a BAM file or a UBAM for unaligned BAM. And then on short reads, we output something called a FASTQ file, which is just like a plain text file. And we can go into the differences later. So yeah, that's at a very high level, that's just kind of what our sequencing workflows look like across all of our platforms. By the way, for this presentation, it's pretty. I think it's going to be pretty informal. Feel free to like pop any questions in at any time. Raise your hand and just ask me anything you wanted more clarifications on. I know software is not everybody's cup of tea, and so any ways we can make this more engaging for you guys or easier to understand, just let me know. Okay, so that's the comparison of workflows. So I'll start with the onso informatics because, like I said, it's pretty light. And so always like to start at a high level and then drill down deeper and deeper. And so for long reads, I'm sure Christy and I don't know, probably uh, who else was presenting? Young, I think today showed you guys like there's a cluster generator and a sequencer, and so you put your flow cell on the cluster generator box to prepare the flow cell. And then when you want to go actually do the sequencing, this is basically what the user workflow looks like. So you, in the lab, you take your flow cell that you've prepared, you load it onto the sequencing box, and then there's two modes you can run here. You can either go into sample sheet mode, where you provide a sample sheet file that contains all of your run settings, and the demultiplexing information like the index sequences that you've attached to each biosample. And then you upload that into the sequencer software along with the flow cell itself. You start sequencing, reads the data from that sample sheet file, and then it knows, okay, I'm going to start sequencing now because you've given me all the settings. And then at the end of the run, it will automatically generate the FASTQ files and perform demultiplexing on the instrument itself. So there's nothing for you to do after that. You just take all the FASTQ files and then you can run them in with whatever downstream analysis pipeline you want to use. There's also a manual mode, and so this is for folks that have their own compute where they want to run the demultiplexing software. And so they can just go up to the instrument. You don't need a sample sheet file. You can just type in however many number of cycles you want to run for this for this flow cell, however many indexes you want to use, and then it'll just do the sequencing, and then you just get a run folder with the raw base calls, but you don't get the FASTQ files. And so you'll take the run folder with the raw base calls and then run another piece of software called OBC to FASTQ on another computer to generate the FASTQ files. And so why would anybody pick one over the other? Sample sheet mode is less hands-on and doesn't really require another computer for you to get data that you can start working with immediately for analysis. But the downside of doing it on instrument is that it takes more time on instrument to run the demultiplexing and FASTQ generation. So you can't start your next run until that's completed. So that could be anywhere between a few hours to half a day, just depending on how much data you have and how many indexes you're using. And so in low throughput situations, this is probably okay. But if you're in a higher throughput situation where you need to maximize the amount of sequencing you're doing on the instrument, then it's probably beneficial for you to just get the raw base calls off the instrument, run the demultiplexing and FASTQ generation on another computer, and then in parallel, just go ahead and start your next run immediately on that sequencer. So just two different options, two different ways for people to run the onso. So sorry, any questions on this?
Can you see the hand raise? Yeah, okay, you got it. I'm dead. You go, Patrick.
Oh, yeah, sorry. Paul, can I ask you a question about the FASTQ structure? I mean, does it have equivalency to FASTQs produced with other short read? Like, can we plug it right into BWA mem, like just some standard pipelines with FASTQs produced by OBC to FASTQ?
Great question. And the answer is yes and no. So yes, it uses, like inside the file itself, it uses the exact same structure. I think I do have an appendix slide on this. Let me see. Okay, so yeah, FASTQ format is exact same FASTQ format that's used by Illumina. I think people just call it like the Illumina 1.8 format or something like that. But what that usually refers to is the quality score encoding that they're using. So we're using the exact same one, and that's been like the de facto standard in the field for quite some time. So yeah, same structure here, same FASTQ, ASCII encoding for quality scores, all that stuff. The only thing that doesn't quite match up with how Illumina does it is the file name itself. And so there's going to be an ONSO update, or I think they're working on an ONSO update to like add that feature in so that you can generate the Illumina style FASTQ file names so that it can be plugged and played into existing short read pipelines that are expecting that Illumina file name. But like it's not insurmountable. Like you could always just rename the file yourself manually to make it work. And not every pipeline requires that. It's just a lot of them assume that it's going to be Illumina formatted in the file name, but that doesn't change the contents of the data itself. So yeah, the answer, yeah, yes, with caveats. I guess the other caveat, yeah, sure. And I guess the other caveat is like there is some quality score calibration assumptions done by some tools, but not all tools, that it's going to be using the Illumina quality score calibration. But like every platform does their own calibration. So if it is making those assumptions, then it's not always going to give you like representative results of what the ONSO quality scores are offering you. But that's kind of like, that's a little niche, more of a niche caveat. But for the most part, yeah, you can just plug and play as long as the file name format is accepted by the tool. All right, great question. All right, going back to here. Okay, so wanted to also just dig into, so we've seen what the workflow, high-level workflow looks like for ONSO, and then just digging into what a run looks like on ONSO. And so we'll start at this level here with you can have multiple bar-coded libraries. Each one has a different set of indexes, and they can be multiplexed into, I don't know, I just call it a library pool. I don't know if this actually has like an official name or anything, but you can pool bar-coded libraries together to multiplex them. Each one of these pools can be loaded onto one lane of a flow cell. On ONSO, there are two lanes per flow cell, and each run on ONSO contains one flow cell. We don't have like dual flow cells or anything on the ONSO system. There's a one-to-one mapping or relationship between the number of pools you have and the number of lanes. And these lanes provide physical separation between the sequenced materials and allow you to reuse barcodes across the lanes, meaning I can use barcode AAA, TTT, CCC, GD, or whatever made up indexes you want, all on lane 1, and then I can reuse the exact same barcodes on lane 2 because they're physically separated, so there's no risk of mixing them and misassigning samples based off of those barcode sequences. There are short read sequencers out there, like the NextSeqs by Illumina. I forget which one. I think like NextSeq 550 or maybe even 2000. I don't remember if they use fluidically combined flow cells, but there are some short read sequencers out there that have multiple lanes, but they're actually fluidically connected to each other, in which case like this wouldn't hold true. But on ONSO specifically, each lane is fluidically separated, so you can reuse index barcode sequences across lanes. So then all of your sequencing settings that you would input into the instrument control software or ICS are applied on a per flow cell basis, meaning if the number of cycles I select for this whole flow for this run would apply to everything in this flow cell. You wouldn't be able to say I want to do a 150 by 150 run on lane 1 and a 300 cycle run on 300 by 300 cycle run on lane 2. It's all or nothing for the entire flow cell. And then after you run the sequencing, it generates base calls for every barcoded library, and then it gets demultiplexed, demultiplexes all of the libraries into separate FASTQ files. All right, so that's ONSO. And so OBC to FASTQ is really the only software that we provide for ONSO at this time that the customer really needs to know about beyond the instrument control software. And so, like I said, this is the tool that is used to convert the raw base calls on that are generated by ONSO, the OBC files, into usable FASTQ files. And so why do we even need this tool? Like, why can't we just use the raw base calls? And that's because the way the sequencer works is that it's taking pictures of the flow cell after every cycle of base incorporation. And so you can imagine this is just like a digital representation of a flow cell. And so you'll have clusters at different physical coordinates on that glass slide, the flow cell. But if I just look at cycle one, like I can't get a sequence from this because this is just the first base of a read across all the clusters, across all the samples. And so what I actually need to look at is just this physical this digital representation of the flow cell across all cycles temporarily, and then stitch them all together. So in this case, I've in this like really dumb mock example, like this is coordinate 1 1 1 2 1 3 2 1 2 2 2 3, etc. I've color coded each of these letters to represent a different sample. So all the red letters belong to the same sample, all the green letters belong to the same sample, and then same thing for the blue. And so looking at this, these are all different clusters on that flow cell. What do we actually need to do? We need to take, okay, cycle 1, A, T, T, C. That's one read for sample 1, the red sample. And at another cluster, I have C, 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 G across these 4 cycles. Then this OBC to FASTQ will pull those out and then write C, 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 G as the next read, and so on. And then it'll do the same thing for the green sample or sample 2, and the same thing for the blue sample, sample 3. So that's basically what demultiplexing is. I hope that was understandable. Any questions on this? Cool. So yeah, that's what OBC to FASTQ does. As far as secondary analysis goes, secondary analysis being anything downstream of the FASTQ generation here, PacBio does not currently provide any secondary analysis tools that are officially supported by PacBio. Like you wouldn't be able to call in to tech support or your fabs and get official support about this or that tool like BWA-MEM or anything. With that said, our fabs folks are pretty helpful. So I think historically they have been assisting wherever they can, but we don't officially support anything beyond FASTQ generation with OBC to FASTQ. And yeah, Patrick, answering your earlier question, we do generate the reads in a standard FASTQ file format, so they can be used with third-party short-read secondary analysis pipelines, although there would have to be like some validation and testing on the customer's part to just make sure it performs the way they're expecting. I have seen some cases where the short-read tools are so used to Illumina data that usually caps out around like Q30 or maybe like Q40 at best. And so they see these quality scores of like Q50 from ONSO data, and it just doesn't know what to do with it. It doesn't accept that value. And so they just abort. And so we've had to like put in requests to have the developers fix them. So that does exist, but that just kind of speaks to the quality of our ONSO generated data, where it's so high quality that some of these tools don't even know how to deal with it. And so that has been an issue in the past that we've worked through. Okay, that's
Hey,
about it
Paul.
for ONSEL stuff. Yeah, sure.
JT here. I just want to double check. I think it was John who asked a question about secondary analysis yesterday. I can't quite remember. This is a chance to kind of circle back to it. We had alluded to that yesterday. Whoever asked the question to Primo, do you guys feel better now about secondary analysis given what Paul is covering? Was it you, John?
Yes, it was. So secondary analysis, the question was, what was the purpose of the secondary analysis phase?
Yeah.
The answer was, it is for applications like resequencing and will automatically do an alignment with the sequence you are comparing your sequence to.
So yeah, I have a more general definition of secondary analysis because we don't like it doesn't always do alignment. I think a lot of secondary analysis is using alignment when there is a reference genome available. But it could be other things too, like metagenomics is not always doing an alignment. You can have assembly algorithms where you don't have a reference genome, so there's nothing to align to at all. And so there's some gray areas around these definitions, but the way we've internally been defining the differences between primary, secondary, and tertiary is on this slide here. So primary analysis is just digitizing biological material into data, just all the processing you do where you don't need to know anything about the biology of the DNA or the genomic material that you've sequenced. So this is things like the base calling, the demultiplexing, the fast key generation, the 5MC methylation calling, the circular consensus sequencing, all that stuff that was talked about earlier today. Like all that is just that's just primary analysis. Like you have these raw sequences, but you don't actually know anything with just this alone. You don't know anything about the biology. And then secondary analysis is more about now you're introducing genomic context, more about the biology into this raw data that you've produced from the instrument. And so, yeah, that's where things like alignment, where you take these raw reads from the instrument and you're aligning it against a reference genome, like against the human HG38 or MM9 for mouse or whatever. And you're saying, okay, this read belongs to came from this loci in the genome, and now you're able to do variant calls to see like, oh, in the reference genome I had this base call, but in the sequenced read that I generated, I see this other base calls that could be a variant call. If you're doing RNA, it could be things like gene or isoform counting. Yeah, a lot of counting type of applications for RNA where you align to a reference transcriptome or even the genome, but you're not necessarily doing variant calls, you're just doing counts so you can do expression, generate expression data to classify, say, like if you're doing single cell, like classifying what type of cell is this just based off of the expression levels that you've sequenced, et cetera, et cetera. And then tertiary analysis is going like a step beyond that. Now typically it's looking at like population level. Like I've generated this data on secondary analysis that gives me genomic or biological context for this single sample. But where does this fit into the overall population? And like, what do we know about this specific variant that we've gleaned from doing a lot of these types of studies on the population in the past? Can I associate this specific variant call to some type of clinical significance, like gain of function, loss of function? Is this associated with a specific disease state? Or yeah, I guess that's the pathogenicity bullet there. So yeah, that's kind of the way we classify the three levels of analysis at PacBio. And this is somewhat standard in the genomics field. You'll see some people call secondary analysis primary analysis at certain places. It's the language is a little bit all over the place, but the way we've historically communicated these classifications of analysis to customers is encapsulated on this slide here. Does that answer your question? Sorry if that was like a mouthful.
That definitely was a lot, but it does answer my question. I have another question
Sure.
specifically about the primary analysis.
Okay.
Will you be going over primary analysis in more depth?
I generally don't go that deep into any specific algorithms for these types of trainings because I think that's like covered a lot more in the technical trainings. Also, some of the stuff I am not super familiar with the intricate details of the algorithms. But yeah, feel free to ask your question. And I don't have the answer. I can like point you to some materials that go into it in more detail.
So, my understanding is the primary analysis on the instrument generates the BAS files, and then post-primary analysis handles the BAS to BAM, the CCS, and the primrose processes.
Sure.
The BAS to BAM is converting the BAS files to BAM files, but what are the purposes of the CCS and the primrose processes?
If I recall correctly, Primrose was a methylation caller. I don't know, I can't remember if we replaced it or not. But yeah, I think that's just one of the methylation callers. So it's just looking at the kinetics data to call like 5 MCs. What was the other one? The CCS that you were asking about?
The
Okay, yeah,
CCS is the second phase of the post-primary analysis.
right. The CCS, let me hold on. Do I have a slide on this? Okay. So yeah, the CCS is, if you'll recall for Revio, it sequences like these smart bells in a circle, right? And it'll sequence that same long insert over and over again. And so it'll generate what's called subreads. And that's just it's sequencing that same region over and over again. So every time it reads it once, that's one subread. And so you just get like this big pile up of subreads. And then CCS, what it's attempting to do is just aligning them against each other, not against like a reference genome, but like all these reads against each other to generate a consensus, because there will be some random errors in the sequencing or the base calling, but the intent is to like by sequencing it in a circle, like circular consensus sequencing, that's what it stands for. By sequencing it in a circle for n number of times after a certain number of passes, if these are all truly random errors and not systemic errors generated by the platform over a bunch of different passes over the same sequence, it should just resolve those errors on its own. Like you should only see if it miscalls a base once, but you have like 30 other calls on that over that same base that say it's a different base, then the consensus should be that the corrected base. And so that's what CCS is trying to do, just correct for random errors in the sequencing by reading the same bases over and over again. So then CCS will convert the subreads into the HiFi. I guess it's not really high. I don't remember what we call it afterwards, but like, yeah, the consensus reads, and then the consensus reads are then passed into deep consensus, which I think Matt talked about a little bit earlier, which runs off of the GPUs that are on the Revio. And so this is just like a machine learning, or like I think it's like a neural network where it's an algorithm that's been trained on a lot of these consensus calls, a lot of data on these consensus calls to find common alignment type of error or consensus calling errors, and then just further polish it. So it's just error correction based off of machine learning. Did that answer your question? Sorry, again, sorry
Yep.
if that's a mouthful. This is also why I don't really try to avoid getting into the details, because it gets really deep for some of this stuff. But hopefully that was a undigestible enough information for you.
That was great.
Yeah. Cool. All right. So then, any other random questions? I can jump around, or we can get back to where was I? Onso stuff. Yeah, I guess that was it for Onso. We've already jumped into the long read side, but I guess we'll start talking about SmartLink here. Okay, everything we just talked about, all this stuff that's Onso, this is completely separate from, at this time, it is completely separate from the long reads informatics ecosystem. So for now, you can just separate these two concepts in your mind. I guess I should have prefaced this whole presentation with everything in this deck is just a snapshot of our informatics ecosystem at this point in time. So like there are new developments always happening. And so like a lot of this stuff, I think even in like the next month or two, we'll have some new announcements and stuff. So like a lot of the stuff will change a little bit. Like we'll add scope and stuff. I mean, in the future, we would love to have long and short reads more aligned together, but at this point in time, this snapshot in time, they are completely separate concepts in terms of like the informatics. So okay, this going into long reads now. So like I said in the beginning of this call, wanted to go over SmartLink, and SmartLink is an essential piece of software that every long reads user needs to have in order to operate their instrument. If you come from a Lumina world, this is basically like base space sequence hub, our equivalent to that. It's used for setting up run designs, monitoring your instrument status, reviewing sequencing results, and you can launch secondary analysis workflows off of your long reads data. If you don't have SmartLink, you can't even set up a run. Sorry, let me move these. You can't even set up a sequencing run. And so this is absolutely required piece of software. It runs locally on premise. So the customer will have to provide their own sequencing computer to install SmartLink server on. And so in this diagram, it just shows like, oh, the SmartLink server connects to your instrument, and you can have multiple instruments connected to one SmartLink server, and it's accessible from just your personal computer. It's like a web server, so there's a URL that's on your local network. So you can just type that into your laptop browser and connect to SmartLink that way. I have some screenshots of what it looks like later. So in terms of sales conversations, when you're talking to new to PacBio customers, you'll want to bring up the compute requirements, and I think I have a slide on that later too, as early as you can in your sales discussions. This has come up in the past where a sale has gone on for quite some time on just the instrument alone, and then at the last minute they realize, oh, actually you need to buy this extra SmartLink server for $5,000 to $15,000, depending on what type of analysis you need to run, and then it just like holds up the deal even longer because there's this additional capital expense that was unexpected. And so, yeah, you'll want to bring this up early in the sales discussion so that the customer's not caught off guard by this requirement for a SmartLink server. Looking at this long read sequencing workflow, it's pretty different from short reads. And so step, I guess, 0 here is you will connect your SmartLink to, your Revio instrument or even your SQL instruments to SmartLink. The user then creates a run design in SmartLink instead of creating a sample sheet, although they can upload a sample sheet into SmartLink. To create a run design, and that run design will be associated to the sample plate barcodes where all your samples are loaded. So then you go over to now that you've set up your run design in SmartLink, you go over to your instrument, you load the sample plate with all these libraries and smart bells and stuff you've loaded that you want to sequence, and you load that onto the sequencer. It reads the barcode and then pulls the associated run design with all the run settings to the instrument, and then you hit go. You can walk away from the instrument at this point. The instrument will start sequencing, and then as it's sequencing, it'll send progress updates to SmartLink. that you can access from a web browser. And then after the sequencer is finished sequencing, then it will transfer all the UBAMs or the unaligned BAMs with the raw reads, the raw HiFi reads to a destination you've set. So it's usually somewhere on your local network, like a storage location. And then once the data's been transferred off instrument, then you can run secondary analysis pipelines in SmartLink, like whole genome sequencing or RNA-seq stuff, metagenomics pipelines that we have baked into SmartLink. Looking at the run structure for Revio is a little different. Oh, sorry, let me go into presentation mode again. Okay, so it's somewhat similar to Onso here. So you have barcoded samples that get multiplexed into what we call a well sample. Each well sample gets loaded onto the sequencing plate in a separate well. So you can have up to 4 well samples on one sequencing plate. You can have 2 sequencing plates per run on Revio. And each well sample gets loaded onto a separate smart cell. So you can have 4 per sequencing plate, up to 8 per run. There's a 1-to-1 mapping between well samples and smart cells. Sequencing settings and analysis settings are applied on a per smart cell basis, very similar to how sequencing settings are applied on a per flow cell basis for Onso. So you can think of smart cells and flow cells as being roughly equivalent concepts. And then after sequencing is done, then the HiFi reads are demultiplexed into separate UBAM files, 1 per barcoded sample. Any questions on this part? Great. So then looking at compute requirements, for SmartLink server, it has some pretty beefy requirements just because of the secondary analysis pipelines. So you'll need at least 64 gigabytes of RAM, 16 CPU cores, if you're doing like whole genome stuff. Local storage, at least 1 terabyte SSD. If you're running... Oh, sorry, this is just for the server part. And then for HPC, the high performance compute, for doing the actual secondary analysis, then you'll need for each node at least 64 CPU cores, and then 4 gigabytes of RAM per core, so 256 total, like 4 times 64, 256 gigabytes total. So this is the part that gets very expensive, along with the 100 gigabytes of SSD local storage for storing all the analysis results. We are working on revised guidance for this, but I checked it. It wasn't quite ready for this presentation. But yeah, I think a new smaller set of requirements that's more budget friendly is coming out. So look out for that. That'll be published into Seismic when it's ready. Network requirements, you'll want at least 1 gigabit copper, optimally 10 gigabits. Yeah, so the cost right now, like I said, is around 10 to 20K for a customer to purchase from third parties. With the lower requirements, it's probably going to be closer to like 5 to 15K, depending on what type of analysis needs they have. So yeah, I don't expect any of you to remember all this stuff, but definitely bring in your fabs, the field application bioinformatics scientists when you're talking to customers about this, and they'll be able to walk the customer through these decision points in more depth. For SmartLink 13.0, which came out last year, there is an option called SmartLink Lite, which is basically SmartLink without the smart analysis. Smart analysis being the secondary analysis pipelines. There are some customers out there that have their own pipelines that they want to run off instrument, their homebrew pipelines, or they take it to like third-party providers. So for those folks, all they need is just like the core SmartLink functionality for setting up runs and monitoring runs and stuff like that. And so the idea is they can install SmartLink Lite instead on just a typical commodity workstation, laptop, or desktop computer. Yeah, we'll see, a lot fewer cores needed, a lot less RAM needed, a lot less local storage. So typically this is like 1 to 2K. And so this could be a good option for customers that don't want to use our smart analysis pipelines.
Would that bypass the customer's need for the HPC node?
Yes, if they don't need ours, let me think. Okay, so I guess there's two customers that I can think of that typically would need this. So one is folks that already have their own compute HPC infrastructure, but it's not using our PacBio provided secondary analysis pipelines. So these are like the Broads of the world. Yeah, they probably already have their own HPCs, so they don't need a special one just for running PacBio stuff. And so all they need for SmartLink Lite is just a separate computer that doesn't that is not an HPC. And then the other customer type is like these core labs where they're not doing any analysis at all. Their customers are sending them samples to get sequenced. All they need to do is just hand off that sequencer in the sequence read outputs to their customers. And so again, they don't need that HPC. They just need, since all the heavy duty processing is being done on instrument, and so they'll take those HiFi reads and just pass them directly off to their customers, and that's it. So yeah. Does that answer your question? Okay, great. Thank you.
Yep.
SmartLink is available for free for download on our website. That comes at no cost. You don't need to log in or currently you don't need to log in or anything. You just go straight to pacbio.com/support/software downloads or click through the nav bar. And then you can just download it and install it yourself. It requires a Linux operating system. There's install requirements there. When should I talk about SmartLink with my customer? Like I said, you should do that as early as possible, but just kind of like walking you through the whole sales, pre and post sales process. Purchase order, while the TAM is working on the opportunity, the FABs or Bioinformatics FAS should be brought in as early as possible to discuss what are their secondary analysis requirements, and they can make that decision at that point, whether they didn't need the full installation of SmartLink and therefore a separate computer to purchase, or if they can get away with just using SmartLink Lite, and so their capital expenses are a lot lower for this sale. And all that, yeah, we should be having as many of those conversations to gather that information in those TAM meetings as early as possible. After the PO is received, then the IT site prep ticket is going to get opened by somebody in the field, so like a field service engineer, a FABs or an FAS. And this is all just to get the instrument set up, make sure all the software is installed, make sure they have like the right computer to install everything. So there's like a whole checklist. At this point, the goal is to, as quickly as possible, close that IT site prep guide, make sure all those those boxes are checked on the checklist, so that installation can be complete. So while installation is the instruments getting installed, folks on site are going to be making sure the SmartLink is installed properly. They'll do a validation run. They'll do the training on site with the instrument once after the software is installed. At that point, you can close out or not you, but like somebody on the field team will close out the IT site prep ticket. And then they can proceed with the wet lab and bioinformatics trainings post-installation, at which point we're now in the post-sales world where the fabs will be on the hook for providing continued support for the software. Any questions? All right.
That's my favorite slide, just saying.
Yeah. Yeah, there's a handful of slides that you should probably you don't need to know all the details, just remember they exist so you can reference them back, and this is one of them. Frequently asked questions. I'm running out of time, so let me like fly through some of this stuff here. Frequently asked questions: what if we don't have the hardware to run SmartLink? You can offer them SmartLink Lite, or in some cases, we've provided SmartLink server loaner computers. You'll have to talk to your fabs about availability of those. Can customers use the cloud instead of buying hardware server? Technically, you can install SmartLink on your own AWS instance. It's not recommended. It's quite difficult. I will say, I think this is not public information to customers, but we've already messaged this internally at like the sales meeting earlier this year, but we are working on a cloud version of SmartLink. Hasn't been announced yet. So that should, once we have that available, that should significantly reduce all these requirements on the customer. They won't have to buy a hardware anymore to connect it. They'll just need an active internet connection with their instruments. But that, like I said, has not been announced yet. So like, let's keep that internal for now until the official announcement later this year. Using SmartLink, I'm not going to go like into details on this, but just wanted to show you like roughly what the interface looks like. So there's like a sample setup page where you can type in like number of samples, number of cells per sample, all that kind of stuff, like constant sample concentration, that kind of stuff. And it generates this protocol that you can print out, and then the wet lab person can follow the instructions for pipetting all the stuff into the right reagents. So then once that's done, to set up, create your smart bells, and you can set up a sequencing run. There's different run setup UIs depending on the different instruments. There's more technical training you can probably watch for more information on this. There's differences in capabilities across different different instruments on what they're able to generate. So Sequel 2 and 2E, they don't have GPUs on board, so they can't do deep consensus. But with Revio, you can, because it's all on board. With Revio, the default output is just the HiFi reads, whereas on older platforms, you will get the sub reads, and then you'll have to run the CCS and all that stuff off instrument or on SmartLink. This is a little outdated because we do support sub reads, but it's like an advanced setting. Anyways, not super important. After the run is completed, you can, or even while it's in progress, you can monitor the run. You can look at the run QC data to see if there's anything off with your sequencing run. It generates a bunch of graphs. I don't even remember what some of these do. They might get removed in the future, but just so you know that these are all here. You can look at the output data, more analysis metrics, things like that. So that's just high level what it looks like. Analysis, I think we already talked about this. This is just, I'm not going to go into high detail or deep details here, but just give you a high level view of like the different analysis workflows we offer for SmartLink. On the SmartLink workflows are a little bit limited compared to what we offer on the command line. So we have some command line tools that are publicly available from, do I have a slide on that? GitHub. So if they want to like more fully fledged end-to-end pipeline that with all the bells and whistles, you can check out our GitHub, github.com/Pacific Biosciences. This will, in order to run these, it requires more bioinformatics savviness from the customer versus the smart analysis pipelines are a lot more click button or push button analyses where you don't need to know how to use the Linux command line and all that kind of stuff. Okay, summary and conclusions. Oh shoot, I gotta go soon. But yeah, so I'm on Dave's call where I have to like give a project update. So I gotta leave. But yeah, bring in your fabs as early as possible. You need make sure customers are aware they need to run SmartLink in order to operate the instruments, and will require hardware purchase for installing and hosting SmartLink. I think that's it. Any last minute questions?